Backyard Green Films is proud to present this episode of Agri-Culture with your host, Alara Bowman. Alara and her husband Rick travel throughout the land in their teardrop trailer that they have nicknamed Maggie, bringing you stories about their travels and the people they meet. They visit farmers, ranchers, and just about anyone who loves putting their hands in the dirt or their feet in stirrups. For the past 3 years, they have been filming a documentary about heritage breed animals entitled The Holstein Dilemma, Heritage Breeds, and the Need for Biodiversity. In those travels, they have gotten to meet some very interesting people. Here's one of those interviews.
Hi, this is Alara. The next interview we're going to share with you is from our trip to Penn State University. We spoke with these two researchers back in November of 2017, almost a year after we had interviewed Dr. Harvey Blackburn in Fort Collins, Colorado. Dr. Blackburn and his amazing work actually came up in our talk with the Penn State doctors, which was a flashback for us, and it was wonderful. Dr. Blackburn apparently has a calf named in his honor at Penn State. So here's the short bio for the good doctors from Penn State University, and yes, this is the short version. Dr. Dechow is an associate professor of dairy cattle genetics. He has an AAS in Ag Science, a BS in Animal Science with honors from Cornell University, an MS in Animal Science from Penn State, and a PhD in Animal Science from the University of Tennessee. And most importantly, he writes some really understandable articles on dairy websites. Dr. Liu is a professor of animal genomics. He has a BS in Animal Science from Northwest Agricultural University in China, and an MS and PhD in Animal Breeding and Genetics from the same. He did his postdoctorate studies at Uppsala University in Sweden, and also at the University of Minnesota. The list of his research studies and publications is a pretty long one, and it uses a lot of phrases like Y-chromosome variations and Y gene content and gene expression. During our interview with Dr. Liu, we found out that China, his home country, has an even larger population of dairy cows than the United States, and demand there is increasing steadily. So his experience in this field really comes from experience in the field. I come from a science family, so I can usually at least understand the neighborhood of a technical conversation. But I was so far over my head I could barely see daylight for this one. Not for 1 minute did I feel that way, though. Here were two guys that had enough letters behind their names to start a dictionary, yet they were some of the most down-to-earth people you've ever met. Both of them are really good at making a really sciencey topic seem like something I have a hope of understanding. Okay, maybe understanding a part of it. Maybe a sentence of it. I got to meet the cows and they were pretty. I found out about Dr. Dechow and Dr. Liu one day when I was knee-deep in research on the Holstein cow and our dairy industry in the United States. I came across an article in the Penn State News entitled "Recovering Lost Genetic Diversity in Holsteins is Focus of Professor's Research." So I read it, and I did some more reading, and then of course we had to go to Penn State. The whole story of what they did kind of reads like a sci-fi movie. 50-year-old germplasm from a long-lost bull saved for the future, thawed and implanted in a modern Holstein cow. The Holstein is often used as an example of what can happen in agriculture when we push for maximum output using intense genetic selection. We find a good trait and we breed for that trait in a single-minded fashion. And increased production is good, right? So how can that be bad? Well, here's what happened. The dairy industry's been doing what all people in their different areas of agriculture have been doing for thousands of years, making improvements to maximize output. Dairy cows are bred for milk production, and when you find one that can really crank out the milk, you mark that one down as good breeding stock. And then all of a sudden, mankind discovers artificial insemination, and suddenly it's the main way to breed many kinds of livestock. And cows are a big one on that list. So which genes do you pick for starter material? The ones that produce, of course, and that's what we did. So now we flash forward to 50 years after AI hits mainstream. The statistics survey at the USDA said that in 2019 there are over 9 million milk cows in the United States. The Holstein breed itself is known to be the world's highest producing dairy animal, and it's estimated to be about 85 to 90% of the dairy cow population in the U.S. Even though the butterfat breeds are slowly gaining ground, we're just a teeny bit dependent on those big black and white cows at this point in our agriculture. and their production volume is pretty impressive. Over the last 10 years alone, there's been an estimated 13% increase in pounds of production per cow. As you'll find out in the podcast, farmers sell milk by the pound, and it's been done that way for a while, but this was a new one to me. Since today's consumer wants things like yogurt and cottage cheese, butterfat's now a big component as well. For the non-dairy people out there, the average milk production per cow in 2018 was 23,000 pounds. So if my math is right, that's a daily average of about 63 pounds, or 7 to 8 gallons per cow. And the average keeps going up over the last few years. These stats are for dairy cows in general, by the way, and Holsteins have an even higher daily output than that. So now back to why they did the sci-fi experiment. Almost all the Holsteins in the United States come from 2 bulls in the 1960s, due to that breed for production thing. Some people say that there are between 16 and 22 genetically distinct Holsteins in North America. Some people say that number is as high as 100. Either way, 16 or 22 or even 100 is a pretty narrow genetic pool to be swimming in for 7 or 8 million animals that are the bulk of our dairy industry. All of those animals have similar genetics and similar breeding patterns and similar susceptibilities. So that's why we called our film The Holstein Dilemma. We have a lot of people on this planet to feed, and we need to produce food at a good clip to do it. That said, maybe it's a really smart thing to have a backup plan for our food production system, just in case something pops up later that we didn't expect. That's where other breeds with wonderfully different genetics come in. They're our backup sci-fi scenario, in case Captain Kirk can't go back in time to bring back the whales. You knew I was going to do a Star Trek reference on this one. We chose the Penn State interviews as our podcast release for Father's Day, as we couldn't resist the irony of 2 fathers, the bulls, having all of those children, and much of the world not really knowing the story. But since we're posting this podcast around Father's Day, I have to mention my dad. Edward Dissler was a surgeon by profession and a farmer and rancher at heart. He had a passion for genetics, beef cattle ranching, packing, and the outdoors. He never looked so happy and fulfilled as when he was digging in the dirt, whether it be with a shovel or on a tractor. When I was growing up, we had chickens, geese, rabbits, sheep, dogs, cats, bees, and a garden that fed the entire neighborhood. The biggest sorrow of my life is that he never had the chance to meet the love of my life, my husband Rick. My second sorrow is that he didn't get to go with us on the wonderful experience that this documentary has been. Tromping across pastures full of hay and manure, standing midfield talking to a farmer, visiting every horse and cow stall at the National Western, analyzing the decomposition rate of the manure we just stepped in, learning how to harness a 6-hitch draft horse team, and riding a mule across Montana would have been the things he would have loved to have experienced. Though now that I think of it, he probably did. No matter how fatherhood came about, in person or not, a happy Father's Day to all. Hi, thanks for meeting with us today. If you would please introduce yourselves and list your titles and honors, please.
Okay, I am Chad Dechow, and I'm an associate professor of dairy cattle genetics.
I'm Y-chromosome Liu. I'm also an associate professor in the Department of Animal Science, work on the animal genomics.
So tell me a bit about your background and tell me how you got associated with this project, please.
So I grew up on a small dairy farm in New York State, and we had mostly Holsteins and a few brown Swiss 4-H projects, and that's kind of what started my interest. I liked showing cows and looking at pedigrees and so forth, and that's what started my interest in genetics, and eventually that led to going to college for dairy science, and that's how I ended up at Penn State, because they had a good football team and that I could watch, and of course the dairy science department.
Most people don't realize it. New York is quite a dairy production state.
Yes, it is. It's one of the top 5 production states in terms of the amount of milk produced in the country.
And what was your background?
So I was born in China and raised in China, got all my education in China. After I received my PhD, I went to Sweden for 4 years. And in 1999, I come to U.S. University of Minnesota, joined Penn State 2007. So my whole life in China, I'm working on the swine part. Come to U.S., work on the cattle, mainly the bovine Y-chromosome. So now I'm working a little bit with the Y-chromosome towards the male fertility, male reproduction.
You both have come to a very specialized arena, both based on bovine genetics. From different perspectives a little bit, maybe?
Yeah, so I don't care about the bulls. They're just because the cows give the milk. So the bulls are a necessary, I don't know if necessary evil is the right word, but they are obviously a necessary component. But we're worried about cows and selecting for cows that are efficient, give lots of milk, but also healthy and fertile and that kind of thing.
So your perspective is obviously a little bit important too,
yes,
right?
I'm more focused on the bull side. I think the male is more important because they impact much larger, much broader.
Actually, that's a really fascinating part of it, because if you could both explain your viewpoint on how the dairy industry has used both bulls and cows differently and how that has directed the genetics that we have in the Holstein industry today in the United States.
So in the past, really genetic improvement comes almost exclusively through selection of elite sires. And obviously those bulls come from very good cows as well, but in terms of how that genetic improvement was disseminated, it's almost exclusively through the bulls.
Yeah, I agree. That's why we're working together on the bull side.
Okay, now it might come from the bulls, but the female seems to be the thing you're really trying to perfect. Is that correct?
Correct. So these bulls, some of them have hundreds or thousands of daughters, and now we even have DNA markers as well. So we determine which bulls have the best daughters based on, we measure how much milk they give, we look at them and see who has the prettiest daughters, for instance, who has the healthy daughters, the fertile daughters. So basically we do a statistical analysis, determine which bulls have the best daughters, and then try to propagate those pedigree lines to the best of our ability.
So the average person might say that's a little bit sexist. When you're trying for the cow, why don't you pick the cow? So if you could explain the logistics of why the bull is the thing that you're shooting for.
From the bull stat point of view, because one bull can fertilize the millions of cows. That's where the male side, why we do the selection from the male side, we put a lot of effort on the male selection because they are impacted much larger than the female.
That's your justification.
That's my personal justification.
So if my layperson's point of view is correct, that grossly impacted the Holstein breed. It's like starting in the 60s. If you could explain how this one track from those two bulls got going here, please.
Yeah, so if we look at today's Holsteins, all the males, if we trace just their paternal lineage, so father to father to father, we can trace them all back to basically two bulls born in the 1960s. And those were, the bulls' names were Pawnee Farm Arlinda Chief and Round Oak Rag Apple Elevation, who was my father's favorite bull way back when I was a kid.
Say that 3 times quickly.
Yes, that's right. But each generation is a subset of the parent generation. And so eventually a lineage gets lost, and slowly but surely we got down to basically just two. There are lots of other bulls in the 1960s that still have a big influence today through female pedigrees and through maternal lineages, but all the male lineages can be traced back to those two bulls.
In the 60s, this is when artificial insemination became king. If you could explain how that impacted breeding.
So really it was starting to take off in the 60s, really the 70s and 80s, it's when it really took off. And so most farms, probably 80% of cows today, dairy cows, are either the result of artificial insemination or at least have the opportunity to be bred artificially. A lot of times, if it doesn't work, a farmer will have a bull running around as well to kind of a cleanup bull, we might call it, to get the cow pregnant if the artificial insemination didn't work. But the vast majority of Holstein cows and any dairy cow in the U.S. is the result of artificial insemination today.
So is this practice just in the United States, or do you have an international perspective?
Yeah, this is across the board in the whole world. And even in China, now they also doing the same direction. I think even the cows using artificial insemination is higher than the US.
Pennsylvania is the place that you both decided to do your study. So is there a reason for that other than the sports team?
Well, yeah, Pennsylvania is actually a pretty major dairy state. We're in the top, I think we're 6th currently for milk production, and we actually have the 2nd most number of dairy farms because our farms tend to be a little bit smaller. So very important dairy state with a very well-established department that's studying dairy science, dairy nutrition, dairy genetics, and so forth.
A lot of agriculture.
Yeah.
Yeah, Penn State is one of the best in the dairy research and study in the history. We are doing pretty well here.
Yeah.
Besides the football team, we are doing very well here.
That other thing.
Yes.
You both have been in the ag department for a while, or did you move into this study, and how did this come about?
I came specifically for dairy cattle genetics. In terms of this particular study where we're looking at the male lineages, that's something that actually kind of started collaboratively between us because Wan Shang's interest is in the Y-chromosome, and we got to talking about, well, I wonder how many Y-chromosomes there really are in the Holstein breed, and well, we better start tracing and find out, and we knew it wouldn't be very many, but 2 surprised us a little bit.
Explain that, if you would, the Y chromosome.
The Y chromosome is so unique because it's only in the males, it's not in the females. So the Y chromosome passes on through father to the son, to the grandson. It never passes through the females. That's the unique part about the Y chromosome. And also because the Y chromosome is, we call the limited transmission. In the male side, it's become one of the best, I think, the ideal part of a genome that can carry the genes that gene may be benefit for the male fitness, if you see that way. Yeah, for the male fitness.
So you're telling me all we really need is the genetics of the male? No.
Yes.
That's such an amazing concept. I mean, we know so much more about DNA, and we're doing switches in DNA, and we're manipulating DNA. I mean, I'm assuming this is something the university really jumped on when you suggested this, or is this something that maybe you just had to work up the food chain when you first suggested it?
Yeah, to me, I think I've been working on Y chromosome for 20 years. And in the beginning, what we realized, the Y chromosome is degenerate, is small. If you compare to the X chromosome, that female have both X chromosome, and the Y chromosome slowly degenerate in the last 200 million years. So compared to the gene content of the Y chromosome, to the partner X, only roughly 3% of genes left. So we would think the Y chromosome was not really important at all. Someone called the Y chromosome a genetic desert, a genetic wasteland. So that idea, if 10 years, 20 years ago, that idea was pretty much people believe now it's changed. Because the Y chromosome, the 3% of the Y gene left for us, they have a much broader expression pattern. They are important.
Okay, explain an expression pattern, please.
So the expression pattern tells us to see which genes express in what tissue, what organ, what time. So the Y genes, most of them express in the testes, in the male sex organ. That's why these genes we think are important for the male reproduction. But what we learn now is that these genes not only necessary for the testes for the male reproduction, but they also relate to the male health in general, like a male cancer, male sex, secondary sex, what's the term we call it?
Sex characteristics.
Sex characteristics for the male side. So the Y chromosomes now have a much broader function than what we believed earlier.
Are there applications for humankind in your studies, in your expression of genes? Are you learning anything that might be applicable to humans, do you think?
Yes, that's very true. For the human side, they now found in the male side, the higher blood pressure, the male certain cancer, they are related to the male testes and the Y-chromosome.
So I'm assuming that this is the purpose of pure research, is finding information to find out how things work, and can maybe be as important as specifically we want to do this one thing, and how do we do it?
Yeah, that's very true. Also for the male side, because in the human, the Y-chromosome tend to have that so-called microdilation. So the microdilation leading to the azoospermia or oligospermia. And that affect roughly 15% of the males in this country, in US. So we do have a direct application on the human side, the human medicine, for the chromosome study, for the Y chromosome, for the paternal lineage analysis.
You coordinate with the USDA's Germplasm facility and say to them, I think this is what dairy cow is important, or how does that work?
Yeah, so they have basically they've collected as much semen as they can from older bulls and from modern day bulls, and so we evaluate that collection and say, are we representing, and they have several different breeds, are we getting a good representation of each breed, and if not, what do we need to do to get that breed represented a little bit better in their collection. And they also, that's how we were able to reestablish some old bull lines, is because they had semen from bulls that were born in the 1950s and that are not related to chief and elevation. So we went back and used that semen to create some modern day calves from those old extinct male lineages.
I can see how genetics from prior years are like a gift from God, so to speak, manna from the sky. I mean, that's something you can't get back once it's gone. Is that correct?
That's correct. If the DNA is not, or the sperm or eggs are not stored, it's lost. So sometimes we overreact a little bit. I mean, sure, we've lost a little bit of genetic diversity in Holsteins, perhaps, but if you look at the global cattle population, there's a lot of genetic diversity in the global cattle population. So we're not at risk of cows going extinct from lack of genetic diversity through genetic breeding. But it is possible that a specific breed could run into troubles because of a bottleneck because of intense selection. So this helps to guard against that a little bit.
So with intense selection, we have, I think someone once said something like 90% of all of our agriculture specific breeds. And other than that, it's, the small farmers and people that choose to raise other breeds. But let's face it, the Holsteins are a good portion of our dairy production. Is that correct?
Yes. So they peaked in the 1990s at about 90% of dairy cattle in the U.S. were Holsteins. That's been declining a little bit. They're now 80 to 85%. And part of that's because our pricing structure has changed, and we now get paid more for the fat and protein that a cow produces rather than her volume of milk. And Jerseys produce a lot of fat in particular, so their numbers are growing. And also crosses among different breeds. The Holsteins were so far out in front of all the other breeds in terms of milk production that crossbreeding didn't really work all that well in the dairy industry for a long time. But now, because we're pricing it more on milk solids rather than just milk fluid volume, all of a sudden those crosses can come pretty close to Holstein in terms of the amount of solids that they produce. And in addition to that, there's some health and fertility advantages to some of those other breeds.
That's the first I've heard the pricing on solids concept. If you could explain that one to me, please.
Yeah, so when a farmer gets a milk check, he's basically paid for how many pounds of protein did you ship to the plant and how many pounds of fat did you ship to the plant. And we're going to make you pay to haul it to us, so all that extra water that you sent us actually counts against you a little bit.
So it's not just straight milk. It is, I need to really microanalyze what's in my milk and the composition. So is this because of nutrition information, or who decided this?
It's more manufacturing. Most of our milk is going to cheese production, yogurt production, ice cream, butterfat. All of those basically have most of the water taken out of it. So we don't need that water anymore. There are some areas where fluid milk is still very important. And in fact, the organic milk market, a lot of that is still drunk as fluid milk, so they are still very concerned with the fluid volume compared to conventional producers who are making milk for cheese.
When you decided to go to the USDA's germplasm facility and say, we need this and this, did you want bulls? Did you want semen? Did you want ovum? How did that work? And did you both have different perspectives on what you wanted?
Well, we were interested in the male lineage and Dr. Liu's Y-chromosome, so we were really only looking for semen.
Okay, so why?
Because embryos would not necessarily, and eggs from females would not have the paternal lineage that we're interested in. So we're interested in specifically those paternal lineages.
Yeah, it's also important to see we want to pick a particular lineage. We want to get that one re-established. That's what we have a much broader opportunity to choose from all the semens.
Okay, so if you could explain to me your study, what you did, you said, I have this idea and this is what we'd like to do, and how did that progress?
So it started, we traced back the number of Y lineages or paternal lineages in the Holstein breed, and we realized that there are really only two, chief and elevation. And then I'm on the Dairy Species Committee with the National Animal Germplasm Program and was talking with one of the scientists at USDA, Harvey Blackburn, about this. And it just kind of, we were brainstorming a little bit and he said, hey, well, let's look and see what we've got in the repository. Maybe there's some other lineages there that are represented that could be reconstituted, and we found two of them, and they're not related to chief and elevation, and we can trace their pedigrees back through the Holstein herd book until about 1880s, and they're unrelated to those bulls through the 1880s. So there are basically two lineages that we found, and we're now in the process of recreating those lineages out at the Penn State herd.
It sounds very much like you're looking to make something for dinner and you go look in the cupboard and you see what you got there before you go make dinner. Is that what you got there?
That was
Sure,
that was right. That's exactly correct.
Right.
Now, that's a really fascinating concept that most people don't know if they aren't in agriculture. I mean, I had no idea of the widespread use of germplasm in our agriculture industry today. It almost seems like that's the rule rather than the exception.
Yes, it is, and there's lots of different reasons for it. One is you don't have dangerous bulls around on the farm that can hurt kids and so forth. That's a big.
Y-chromosome is a pain, isn't it?
Yeah, well,
Yes,
I don't know
right.
about that. Has to be properly
Yeah.
managed, that's for sure.
Yes, I do.
All right.
So in this study, then you said you're going to do what? We have the germplasm.
So we're going to study the fertility of these bulls from different lineages to see, have we lost some genetic diversity that could be beneficial in terms of improving male fertility. That's the first step. So we'll collect some semen from these bulls, and maybe in time, because we haven't just lost diversity on the Y chromosome and for male fertility, but we've lost genetic diversity that relates to lots of different traits. As we've intensely selected, we've got really high milk yield, fat yield, protein yield, but our fertility levels in our cows have slipped a little bit, for instance, and there's maybe some loss in some cow health characteristics. So in time, we may be able to reincorporate some of that lost genetics back and recover some genetic diversity that would be really beneficial to modern day farmers. But that's a little bit of an unknown and will take a long time.
So when you say genetic diversity in Holsteins, it seems almost like a contradiction of terms in a way. But I actually think the breed might be a great example of engineering for a purpose, so to speak. And by engineering, I mean selecting animals to breed and what to cull, and also the positive aspects of bringing in material within the breed, but from an outside line. So would you kind of talk about that, please?
Yeah, so there are probably 6 to 7 million Holstein cows in the United States. So even though we use a lot of AI and do intense selection, just because there's so many cows, there's going to be lots of genetic diversity, even through just mutations that occur each generation. So there's still a fair amount of genetic diversity in the Holstein breed that can be capitalized upon. But we have lost genetic diversity through intense genetic selection, and some of it were family lines where maybe they had some good genes, but they didn't really have enough good genes for us to really select them and to keep that line propagated. And now we're starting to look back and say, okay, can we incorporate some of those good genes that were lost back into what we have today to kind of maybe get the best of both worlds? Cows that give lots of milk and fat and protein are fertile and healthy and have no difficulties giving calves and so forth.
Now, this might be a sensitive topic, but I've heard statistics tossed around that all the Holsteins in North America are basically related to something like 16 cows, or some say it's a genetic pool of about 16 or 12. Is that a truism or no?
It's a little bit exaggerated. Geneticists calculate something called effective population size. And the effective population size it's less than 100. And basically what that means is that if we had 100 cows, we could put all the genetic diversity in the Holstein breed into those 100 cows. And that sounds like a really tiny number, but it actually represents a fair amount of genetic diversity. 10 would be a really small effective population size. And there is concern over the last few years due to genomic selection technology that our genetic progress rate is speeding up even more quickly. So how much will that effective population size drop even further? And we probably are pushing 50 now in some instances, and so it is dropping, and it is becoming a concern. But again, that's a concern for the Holstein breed, not cattle in particular. We're not going to make cows go extinct.
You talk about the effective population. So for the female side, we said the Holstein now totally have about 94 below 100. But the female side versus the male side, the male side only 2. That's what we decided to real. The male side we knew is only 2. For the female side, you put them all together, it's now about 100.
That's a very interesting point, because 100 females, but 2 males that have some fairly important Y switches, that's a concern, is it not? Because those Y switches, if they have something that brings the characteristics forward, I would think that's a note of caution, even if you can mix. I can see why that third bull is really looming on your critical horizon, is really important to your study, no?
Yes, I think that's yes to my answer is to a certain extent. So the 2 bulls for the genome diversity or genetic variation, besides the Y-chromosome, all like the female side, should be the same. With only the male side for the Y-specific region, their part of variation diversity is very much limited. We still try to understand if the 2 bull, even 1 bull, can literally survive for a breed. We don't know that. We try to understand that.
So the counter argument is that if these bulls had serious defects on their Y chromosome, they wouldn't be fertile enough to have taken over and represent so much of the population. So we have a little disagreement about how important is that lack of diversity. It depends on your perspective. But certainly there is limited opportunity to make improvements in male fertility. So our male fertility currently is sufficient. If we wanted to make it better, our opportunities to do that are probably limited because of the lack of genetic diversity.
So it's actually a good thing, I would think, that you have this Holstein that seems so secure, but what if we could make it even better and more secure? It would be excellent, especially when our food supply is so heavily dependent on these animals.
Yeah, and I would say that the Holstein breeders in particular, they want to maintain the purity of their breed. I mean, that's part of being a breeder. And so the simple solution is that if we run into a problem with Holsteins, then we just cross Holsteins with Jersey or Brown Swiss or something like that. But as a breeder that's dedicated to maintaining the purity of your breed, you don't want to do that. So finding additional lines that can help you to maintain the diversity you need in your breed without compromising and bringing in genes from another breed is really important.
So if you lose 20% of your production to do that, that might be a really good deal.
Yes, that could, yeah.
With your study, you got the germplasm, and you did what with it?
So a company called TransOVA Genetics in Iowa took the semen and then they aspirated some oocytes from some elite donor cows and created some calves in vitro, and then those were implanted out in the Penn State dairy herd, and there were 2 different bulls. Calves from the first bull were born in March, and calves from the second bull are going to, I think 3 of them have already been born, and there's about 6 more to be born here this month, November.
Do you get to choose if they're bulls or if they're male and females when you get the calves, or is it what comes out, comes out?
What comes out, but that wasn't our original plan. So that was one of the little hiccups in this project. So the idea was that we only really wanted the bulls, so we were going to sex the semen and only create male embryos. But when TransOVA went to do that, they found out that old semen from the 50s was a little too fragile to sex. So they just had to create embryos. And so it's a little bit of a surprise what ratio will come out.
And you were hoping for the males, I take it.
Yes, I'm only for.
I'm hoping for the males too for this project, yes.
So there's a couple different batches here. And is this from the 2 bulls and then the third as well, or is it just the third bull?
Just 2 bulls.
Okay, so there's 2 original bulls.
Oh, there's 2 original. So these are 2 new, unique lineages that are not related to those originals.
So in total we got 4. The 2 plus these 2.
So Wan Sheng's sample size for bull fertility has gone from 2 bulls to 4. We've doubled his sample size. So
Yes.
I looked at the pictures in the Penn State article that came out. Do they look the same as the Holsteins that you've come to know and love, or do they look different?
they look different. They're fatter and sturdier.
I did not get a chance to really check that. Here's the one that managed to.
Fatter and sturdier, but markedly so. The way you look at them and say, wow, that's a different animal.
Yeah, maybe not a different animal, but certainly not near as lean. We've selected those modern day Holsteins. Their job is to produce milk and not meat. That's what Angus are for. So they are really genetically programmed to grow big and lean and produce lots of milk. And these guys from the 1960s have a little more meat on their bones.
You've got a couple batches here, and then what's the next step?
So these bulls will then be taken to select sires, and semen will be collected from these bulls, and lots of information about the fertility of those bulls will be collected and the semen quality. And then Dr. Liu will study how their fertility and semen quality differs between chief and elevation lines. And then there will probably be some calves created from those bulls as well, just to see how calves from those old genetics perform in a modern day environment.
With the new bulls, will you be donating some of that back to Dr. Blackburn?
Yes, that's a very important part of this project is, yes, so we will donate semen back to the Germplasm program. And so it's an attempt, and that's why we used really modern day females to mate to these older bulls. So it's an attempt to try to update that really old lineage a little bit. So we're maintaining some of that lost genetic diversity on the Y-chromosome. So we're reintroducing that into a lineage that's more representative of modern dairy cows.
Because it's very much like a library, is it not? You can have 50 different books, and as long as the books are there, you can always go take them out. But if the book is not there, more like an encyclopedia series, I guess would be a better example. If you have 50 copies of the same encyclopedia, it's not as effective as one copy of each volume of one set, because you can always go pick a different book. There's still 50 books, but it's a different book.
Right, yes. Correct. So we want, so if we get a little more diversity, it gives us more opportunities to make some changes.
With Dr. Purdy, we got to see him do the actual testing. So he's studying for motility, and are you doing that type of thing, or do you have to see the traits actually express themselves somehow?
Yes, we study from their semen, the sperm morphology, the sperm motility. So that's from the semen quality stand of view. We also study their genome sequence. When this animal, now we collect the blood, we sequence the DNA for the comparison of this animal with the current Y chromosome. So we can do two different levels. One is the phenotypic level, the other one is the genetic level.
So once you have this, then you're going to continue to breed and see what comes out down the line, or how does that work?
So we're going to do the fertility studies. After that, we don't really know for sure. We'd like to see that happen where, so we continue to maybe breed these bulls to another group of elite females and try to slowly incorporate that lost genetics back into the Holstein breed. But we've made so much genetic progress since 1960s that they are so far behind in terms of how much milk we expect our daughters to produce that it's a little bit, it's an unknown. We'll see how it works.
As long as you have the gene, this is not as fragile as the 1960s bull. So a couple of generations down the line, if there were genes that you wanted to express in the daughters and still maybe maintain that milk production, it's possible that in 20 years you might be able to switch which ones you want, which ones you don't, correct?
Using some gene editing techniques and CRISPR and those types of things, yes, theoretically. We have not really, we've used that type of technology experimentally for things like some cows are born without horns, so we can overwrite the horn gene and have calves that are born without horns. For things like milk production, because it's influenced by so many genes, there's not some magic milk production gene. It's the entire genome that's determining how much milk a cow gives. It's going to be a little bit trickier. And there are also regulatory issues that need to be sorted through about, is this a GMO? Do we want GMO animal production and so forth? So it's a little bit of an unknown in that area.
Yeah, I guess I agree with that assessment. We are not really sure if these males come in, if the industry is willing to accept, because the difference between now and back in 96, the genetic improvement in the past. So that's a concern, but we will see how this come out with these bulls, with their offspring, particularly in the reproduction side, how much difference between this group and the current population.
When it comes down to it, we truly don't know exactly what makes every single thing tick, do we? We had the opportunity to speak with Temple Grandin, and she was saying that when they selected in pigs for a specific production characteristic, at the 5-year mark they discovered that a Hock disorder had cropped up, and they never knew that the one thing they were selecting for turned off a different thing. It just switched a different switch. So then they had to back into the genetics to try to correct it. Have you found anything in your studies or in your experience that you thought was one switch, but that it turned on a different one that you didn't expect?
So you might want to talk to the Penn State Ivanhoe.
Penn State Ivanhoe star. So that's a really excellent example. Hanging on the wall over there is a picture of a bull named Penn State Ivanhoe star. And he was born in the 1960s as well. And he actually does have at least one male descendant left. So it's not just Chief in Elevation. Penn State Ivanhoe star still has one out there, but out of thousands. So there was a bull named Penn State Ivanhoe star who was really well known because he had daughters that produced a lot of milk and had the proper conformation, looked good, had nice udders and so forth. And he had a son that was even more famous named Carlin M. Ivanhoe Bell. And his daughters really gave a lot of milk and was used all over the world. Well, in the mid-1990s, some farmers were starting to notice, I've got some calves that are born and then they die pretty shortly after birth. What's going on here? And it turned out to be an immune system problem called bovine leukocyte adhesion deficiency. And these calves basically didn't have a functional immune system. And they traced back, and what had happened was that Penn State Ivanhoe star had that genetic defect. And he propagated it to his son Bell, who then spread it around the world, and then eventually we ended up with calves that had 2 copies of that defective gene, and they ended up dying. And then shortly after that, within 10 years, we found another genetic defect called CVM, complex vertebral malformation, which resulted in stillborn calves. And that happened to also trace back to Penn State Ivanhoe star. So he's a really famous example of, you get these famous bulls spread their genes all over the place, and then we find out, there was a problem there that we weren't expecting.
That happens quite a bit in science. If you think about DDT, we thought it was lovely and wonderful, and then all of a sudden, 50 years later, there's all kinds of stuff that pops up. Does that scare you at all, or does that make you cautious as scientists?
It does. Yes.
Yes,
and especially in the faster you make genetic progress, the faster you could potentially be propagating genetic defects. But the good news is with a lot of our DNA technologies and genomic technologies now, we're also better at finding and eliminating those than we have been in the past. So it worries me, but it's not something that I'm so worried about that, you know, we need to stop making genetic progress because something bad might happen. If something unfavorable happens, we'll find it, we'll eliminate it, and we'll move on.
Because the reality is, in all of these things in genetics, like your poor bull that introduced everything into the Holstein, once it's out there, it's very difficult to get back.
Yeah, well, with genetic testing, that would certainly be true with GMOs. Once it's out there, it's going to be very difficult to get back. With a genetic defect, we can now identify it, and essentially by making sure we're using bulls that don't carry it, we can maybe not completely eliminate it, but get it to such a low level that it's inconsequential.
But this is with something that's a birth defect. You know, if we had something like an immune response that we didn't necessarily figure out until 20 years later, that might be a concern. Who knows? I mean, who knows why colony collapse disorder occurred? Might be something 30 years ago, might be something now, but it takes a while to figure that particular thing out.
So that's the reason, the main reason you guys do this for this preserve the genetic diversity, I guess, yeah.
That would
provide the genetic, yeah. The genetic diversity provided the...
Right, exactly.
Yeah.
So it's not that one way is the perfect way for everyone, but if you have that bank there of genetic material in Fort Collins, you can always call them and say, I need that material.
Yes, I need to go back 50 years. That's what we are doing now.
Yes, that's right.
Go back 50 years to get a new Y-chromosome out.
Exactly, and if it's not there, you can't do it. The argument might also be if you have cows on the ground and they're breeding to adapt with today's changes, you know, climate, disease resistance, all of those things. And if you pull something out of the freezer that doesn't have today's resistance, are you introducing susceptibility in some ways?
That's possible. There may be some genetic defects from long ago that didn't really get propagated, that exist, and that could be spread because of that. I actually tend to think, though, I'm not too worried about just in general adaptability and so forth from those older lines, because as we make genetic selection, we're moving that animal further and further and further from the natural state. And in the natural state, they're optimized towards high survival, towards healthy offspring and so forth, because we don't have an artificial environment where we're going to give them antibiotics if they get sick and so forth. So the further we move from what God and nature perfected, the more we should expect challenges, poor health, fertility challenges to come along.
What do you see for the future of your study? What would you like to see?
Okay, so for my study, with this study we collaborate, we will hope to get this new Y lineage or male lineage being adopted by the industry. We discussed that earlier to see we have some concern on that, but this is something we try to see how that works. For a bigger picture, we think the most contribution from this new line is the genotype that 30, 40, 50 years ago that may include traits like reproduction and others that have been lost in the past 50 years. So can we bring that back to the industry? That's something we are really looking for.
But that's actually what excites me about this particular study is that with cattle we have, because we've collected semen on all these bulls over many generations, we have really kind of a unique opportunity to study how the Y chromosome changes. And that to me is more interesting to some extent than adding genetic diversity back, just because that's easy to do and we'll learn a lot. Adding genetic diversity back is harder to figure out how that's going to be done.
Put the puzzle together rather than take the puzzle apart.
Yes, exactly.
Yeah. But either way, I would think it would be really fascinating in terms of the information that you can glean.
Yeah, so that's
how the thing functions.
Yes, that's what I'm excited about, is generating this information and then letting Dr. Liu do the genotyping and tell me what it all means.
Do you think that the average consumer understands that the cost of the milk or the price of a gallon of milk that they're paying might be dependent on the USDA funding for a project like this?
So funding for ag research Actually, I don't think it's been cut in the last few years. It's actually growing.
It's
USDA funding maybe.
But the USDA funding is a very small, only 2% of the USDA budget used for research. And in the past few years, it's keep that 2%, but you can see inflation and everything. So my feeling for my research, the USDA funding is down to a level we could not tolerate.
Yeah.
They need to keep the priority, keep the research going for this country. If U.S. want to have a competitive agricultural, livestock industry to the world, they have to.
It's interesting in the Articles of Confederation. It didn't make it to the Constitution, but in the Articles of Confederation, some of the purposes of the federal government were to support science, scientific research, and the arts, because those are things that are hard for a company to really. I mean, obviously, companies do a lot of research and development, but a lot of basic science, if they don't know what the end result's going to be, it's hard for them to justify researching it. So that's where the government funding really becomes really important. In agriculture, the margins are so tight that there aren't very many companies that have enough money to really invest a lot in research and development. So it is really important, and most of the advances we've made in agriculture, it goes back to government-sponsored research to some extent.
Would you give me a blurb and explain how you take something out of the freezer and make it a living, breathing animal?
Okay, so what we did was we took semen from the national animal germplasm. We shipped that to Iowa to TransOVA Genetics. They took some cows that were owned by a company called Select Sires Incorporated, and they went in and they collected eggs from those cows. And then they did in vitro fertilization with that really old semen from the 1950s and those eggs from modern day cows, created embryos that were then implanted into surrogate cows out here at Penn State. And those calves were born in March, the first batch, and we have another batch born this month.
That's Blade Runner, isn't it?
Yeah, so.
Yeah. I mean, that's sci-fi. It used to be, and now it's reality.
Yeah, that's
yes.
right.
So what's the outlook in the present and in the future?
I'll let you go first.
No, I'm
One of the ag departments.
Yeah.
no. So
Free rules.
yes, our Department of Animal Science is getting stronger. It's doing good. We merged from the poultry science. We now have 35 faculty members. We're doing good. So research-wise, we're doing good. I think we were getting better. We are Penn State.
I think it's an exciting time to be doing research in animal genetics just because we have all these new genomic tools that are available now, and so it's keeping us busy. And we're also seeing diversity in terms of consumers and what they want. We've got consumers that want organic products, consumers that want local products, and that actually drives a need for genetic diversity to some extent too. So it's an exciting time to be a dairy cattle geneticist.
So as usual, we got some production shots with the good doctors, and they gave us a little tour and told us stories about the decorations on the walls of the room we were in.
Okay.
I'm going to stand in the midst of the. I'll be the X in the middle of the Y's.
So these are 1922.
1922.
The ideal cow in 1922. The ideal cow and bull in 1922.
And today?
This is the ideal cow today. So you can see
How do you know it's 1922? You got a date
ribs.
somewhere?
Look at the ribs on this cow.
Yeah, we want big open ribs because that means that there's no fat over those, so she's not wasting any fat. She's putting it all in the milk pail. And you can see the udder up here tilted a little more. And
He's
here's
a
higher, wider
over
rear
there.
pail.
I'm trying.
Oh, that one looks so much. I'm German,
Yes.
so maybe that just looks healthier for me because there's more fat on
Well,
it.
she probably is a little healthier, but she gives more milk.
Okay.
All right.
Okay,
I'll
wonderful.
send you those pictures.
Okay.
Thank you
you
very
so
much.
much. I appreciate
I really
it.
Well,
appreciate it.
Thank
you.
you.
This is delightful. I love it. At this point, Dr. Liu had to leave us, and Dr. Dechow took us out to see the calves.
Right now we are at the John Almquist Center, which is where a lot of pioneering research was done in the artificial insemination industry. And we've got our 3 special bull calves here that are from the old genetic lineages. And so we're here at the Almquist Center right across from the Penn State football stadium.
Well, they're definitely a little heftier, aren't they? Hi, guys.
So these 3 bulls are a cross of some modern day genetic line Holsteins with a bull from the 1960s era that was actually bred at the University of Minnesota. So that bull's name was Cuthbert, and he was born as part of a selection experiment that was conducted at the University of Minnesota, where they wanted to compare 1960s genetics to modern genetics. And what they did was they split their herd in half, and half of the herd they bred to the very best bulls of the day, and the other half of the herd they just bred to bulls that were born back in the 1960s, just indefinitely using semen that they had collected from that time period. And so their dad was born as part of that selection line that represented 1960s genetics. These old genetics are supposed to be a little bit smaller in terms of be shorter, but we think that they are certainly carrying a lot more body condition, a little bit more heavily muscled than what we would expect from a modern day genetic bull. And we'll see, their daughters would be expected to give less milk than a modern day Holstein. They do have some sisters up at the dairy herd, so we're going to track and see how well their sisters milk, and then when these guys get to sexual maturity, we'll collect semen from them and just test and see how their semen motility and fertility compares to that of a typical bull, modern Holstein lineage. Bull on my right, your left, I guess that would be, is named Harvey. So he's named after Dr. Harvey Blackburn from the USDA. And then the other two bulls are named for some dairy scientists at the University of Minnesota who are involved in the long-term selection project that created their father.
Is this the equivalent of the astrophysicist naming a star after them?
Yes, that's exactly.
Except the stars don't get slaughtered afterwards at some point, right?
That's true.
So they definitely look a little meatier to me.
So the big differences would be from the 1960s era to today, that those cows in the 1960s were giving about half the amount of milk that today's Holsteins would be. They look quite a bit different. They're shorter, they're sturdier, they carry more muscle and more fat. Sometimes we talk about we've selected cows to give a lot of milk, and that's true, but that's not the only thing that we've improved. The udders of the cows have changed a lot. So to get all that milk out of those cows without giving the cow increased chances of having udder infections and so forth, we need to make sure they have lots of volume, but we don't want the udders dragging on the ground. So we've selected really intensely to have udders with really good conformation that are held up high and tight to their body with big, high, wide rear udders and so forth. So the udders of these bulls' sisters we're expecting to be not as fancy as a modern Holstein cow would be. So they'll give less milk and they won't look quite as pretty, but they'll be sturdier and more fertile is the expectation.
So how long before you can get enough successive generations? I mean, I think cattle have a 9.5-month gestation period, and you breed them at 20 to 24 months or something. What's the?
They would first have their first offspring at 20 to 24 months.
Okay.
Yeah. 2-year generation interval. So they're sexually mature when they're about a year old. So basically 2 years is about how quickly we can turn over our generations. It's going to be tough though, because we've made so much genetic progress in 60 years. These guys are so far behind, even though their mother was a really elite cow. For them to have offspring that are going to be able to compete in a commercial environment, it's going to be tough, so we'll see.
But is that the purpose of them?
Not really. Their purpose is just to understand more about Y-chromosome variation, fertility, and what sort of other types of genetic variation that's been lost that maybe we can hope to recover.
This is your backup drive. You might not use it, but it's there.
Yes, that's exactly right. Well, we will use it to do some basic scientific research. Whether it has a commercial production utility down the road or not, that's something that we don't know.
If you liked our podcast, please subscribe. This is how we keep going. And please tell your friends to join us.
We want to thank Dr. Liu and Dr. Dechow for being kind enough to meet with us today. If you would like to find out more, please visit Penn State University at www.psu.edu, Agricultural Science section. Also, the National Dairy Council is found at www.nationaldairycouncil.org. Thanks again. You have been listening to Agri-Culture with your host, Alara Bowman. Please tune in for more upcoming episodes from our travels. We'd also like to thank our producer, Michelle Council. I'm Rick Bowman, your behind-the-scenes editor. Until next time. This has been a presentation of Backyard Green Films Productions, all rights reserved, copyright 2019.